The last class we talked about how the nervous system works in terms of how an action potential is generated, what's happening with the movement of ions, and then talking about the resting potential where nothing's happening in the nerve fiber, moving on to the generation of an action potential, that is the nerve impulse that is transmitted along the nerve fiber, how that happens, and then talking about what happens after that, so how the resting potential is recovered and the refractory period. Today we will take a look at uh, how the impulse travels along, right? So we've talked about how the impulse is generated. Today we'll talk about how the impulse travels along the nerve fibers and then the neurons. So this is chapter 8A3. The neurons in action. So what happens when an action potential is generated in response to a stimulus? And so it's not generated immediately throughout the length of the fiber. It generates at one point, the starting point, and then it travels. So if this is your nerve fiber, and the impulse will start from this point, obviously, right? It starts from here and then it travels along, and so somewhere over here it's moving forward, right? And so there are changes happening at 3 points. Point number one is where the action potential is, point number two is where it's going forward, and point number three is what's happening to the region that it just moved ahead from. So if you look at point number one, right, that is where the action potential is. Right, at point number two, the action potential is being set up. Right, so for it to move forward. And I'm talking about this region so it makes it easier to understand what's happening behind, because if I talk about this region where it's starting, then I can talk about what's happening when it's moving forward, but there's nothing to talk about before. But these 2 points are applicable over here as well. The action potential is at this point, it has already been generated, and at point number two it is being set up, and then we look at point number three, and so that is undergoing the refractory period. So this also tells us that it's not that the entire length, I mean, it's happening very quickly, it's happening in milliseconds, right? The nerve impulse is it's traveling the entire length of the fiber within milliseconds, and that's why you have anywhere between 500 to 1,000 impulses traveling per second. So it's happening super fast, but even then, right, it's happening in steps. So as it's moving along, it is generated at one point, it is setting up the impulse ahead of itself, and the region before is undergoing the refractory period. So it's not that the whole fiber has the action potential at the same time and then the whole fiber is undergoing the refractory period at the same time. That's not how it's happening. Again, even though it's super fast, it's happening in steps. So for this, I will take you to the book because the illustrations are good and they will help me explain rather than having to draw all of that over here. So as you can see over here, the illustrations from the book, and they explain it very well. So this is step one, not the step one that I've drawn in, on the whiteboard. So this is step one, and this is the fiber at resting potential, right? So nothing's happening. And so at the resting potential, there is a positive charge on the outside and negative charge on the inside. We know that, right? With high sodium ion concentrations outside and high potassium ions inside. Okay. Then an action potential is generated. So when stimulated, voltage dependent sodium ion channels open, sodium ions flow in, the axon is depolarized. We know that, we've talked about that. Localized electric currents are generated in the membrane. Okay, so this much we know. Now, what happens after that? So that's step three. And so the potential difference in the membrane adjacent to the first action potential. So this is where the first one was generated, the dark purple. It was light purple before. And that causes a depolarization in the adjacent area, which is now light purple, as you can see, and you can see the movement of the ions, right? In step two, this region had sodium ions coming in. In step three, now it is undergoing refractory period, the refractory period. So the sodium ions and potassium ions are being pumped out, and the adjacent area has sodium ions being pumped in, and so that is having the action potential generated. So a second action potential is initiated. That's the light purple area. And at the site of the first action potential, the voltage dependent sodium ion channels close. So they're not allowing any more sodium to move in. And the voltage dependent potassium ion channels open. So that's the refractory period where they're pumping everything out. Potassium ions leave the axon, repolarizing the membrane, and the membrane becomes hyperpolarized, the overshoot that I talked about yesterday, right? It's also called hyperpolarization. And then step four, so this is showing all three, right? So you have, right, this part where sodium ions are moving in, so that is where the action potential is. This part where it is being hyperpolarized, and then the first part where it is going back to the resting potential. So a third action potential is initiated by the second. In this way, local electric currents cause the nerve impulse to move along the axon, and so this is the the direction of the impulse, the arrow. At the site of the first action potential, starts back here, the potassium ions diffuse back into the axon, and so the resting potential is restored. So this is how an action potential travels. Depolarization causing depolarization next to it, that starts going into the refractory period, and then right as it travels along after the refractory period, after the overshoot and the hyperpolarization, it goes back to the resting potential. So it's happening in steps, it's happening sequentially as the potential moves along the nerve fiber. Is this clear?
Yes, sir.
Okay, so let me just move back to the whiteboard. So let me just go back and add, right, hyperpolarization over here. So it is hyperpolarization. Okay, and so with that illustration, we've covered this. Next, we want to talk about saltatory conduction. Right, and this is how the impulse travels in myelinated neurons. Right, where the action potential seems to jump. Right, so if you remember the myelination process, there's the myelin sheath that's wrapping around the nerve fiber, and then there are intervals where there is no myelin, and those are called the nodes of Ranvier. And so the action potential jumps between the nodes of Ranvier. Why? Because the myelin is the cytoplasm of the cell and the cell membrane, right? And the cell membrane is phospholipid, so it's lipid, it's fat, right? And so that's going to insulate the nerve fiber. That's the point, right? And so the ions are unable to move in or out in the region that is covered. So ions cannot move across the membrane of the fiber through the myelin sheet. And so they can only do that, the depolarization, all of that can only occur at the nodes where there's no myelin. And this helps in speeding up the impulse, right? The reason why it helps speeding up the impulse is because now, right, the exchange of like the movement of ions doesn't need to happen through, along the entire length. So, right, movement of ions does not need to occur along the entire length of the fiber, right? It only occurs at the node, so that saves time and resources, and that speeds up the impulse. Right, and so yeah, that's all there is. There is an illustration in the book to show this. You can take a look at that. It's showing that basically the action potential is being generated only at the nodes and not the regions where the myelin is. They mention a tip, right, as a clarification. So saltatory, right, is taken from a Latin word that means to jump, and that's why we're saying, right, they're jumping, but they're not actually jumping, right? Basically, what's happening is that the local currents are spread or the term they use is elongated. Uh, to start depolarization at the next node. Any questions?
No, sir.
Okay. Next up are synapses. So I think I'll move to a new page for that. right? And these are junctions between neurons, right? And this is from where, right, an impulse is transferred from the receptor to the sensory neuron to the relay neuron to the motor neuron to the effector, right? So at each of these steps, and basically, right, between the relay and motor neurons, the sensory relay and motor neurons, but right, you got synapses, and so they are what bridge the gap when impulses are transferred along these neurons. So one is the impulse transferring along a single nerve fiber, and then the impulse is that's the impulse traveling along the nerve fiber, and then the impulse is transferred to the next fiber, whether it be a relay neuron or a motor neuron or an effector or whatever it is, right? And so that particular structure is called a synapse. So what does a synapse include? It includes the following parts, right? So it has a pre, sorry, not per, pre synaptic knob. The reason why it's called a knob is kind of because of its shape, right? It looks like a knob. Then you have the synaptic cleft, which is the space in between. And then you have the post synaptic knob, right? These are the structures that make up the synapse, and then along with that you have the neurotransmitters and the receptors for those neurotransmitters. So different neurons don't touch each other. They are very close, right? So the synaptic cleft is a very short gap. And the reason why it's very short is to make diffusion efficient. So we know that diffusion is only efficient over very short distances. And so the neurotransmitters, as we'll talk about, travel from the presynaptic knob to the postsynaptic knob through the synaptic cleft via diffusion. And so the shorter the gap, the more efficient it will be. But point is that neurons do not touch. So it could be very close, but still not in contact. And so this part is a synaptic cleft, this is the presynaptic, and this is the postsynaptic. Okay. Now what you have is when the impulse reaches the presynaptic knob, right? It changes the permeability of the membrane. Of the membrane, the presynaptic membrane, to calcium ions. Right, so I'm writing the the formula for the symbol for the ion, but calcium ions. And it does that by opening up calcium ion channels. So calcium ion channels open, and calcium ions enter the the fiber. They flow in. Right, and so there's a concentration gradient, obviously, so they're flowing in, their channels, so diffusion's happening. And what that does is it causes, there are vesicles that contain the neurotransmitters, right? And those are, caused to move towards the presynaptic membrane because of the calcium. So the vesicles fuse with the membrane and release the neurotransmitters into the synaptic cleft. And so each vesicle has about 3,000 molecules of the transmitter. About 3,000 molecules of the transmitter. Now, in the synaptic cleft, whatever the neurotransmitter is will diffuse across, and binds to receptors, right, on the postsynaptic membrane. And what that does is that it initiates an action potential. In the postsynaptic nerve fiber. Now, I said that it initiates an action potential. That is the case, right? So the sodium ion channels will open and sodium will move in, etc., etc. And that would be the case if the neurotransmitters are, you know, causing an excitation, right? And so that would be an excitatory postsynaptic potential. And so that's EPSP. Right, and so the whole concept that there's a threshold, and so if it reaches the threshold, it will go all the way, and the action potential will travel along and it's fired. So all of that we've talked about. Now, there are some cases where the neurotransmitter does not cause an excitation, it actually has an inhibitory effect, right? And so in that case, right, so what's happening over here is, it's moving from -70 to +40, and so, right, that's what's happening. But right, in case of an inhibitory, so so if the neurotransmitter causes an inhibitory postsynaptic potential, and so that is IPSP, right? And so what that does is that, you know uh, further polarizes, it makes the inside of the fiber further negative. So what this does is it, some different, not the sodium, but different ion channels open, allowing, right, negatively charged ions to move in, and that makes the inside more negative. And so from what we've understood so far, we know that an action potential is not going to be generated. In fact, it's going to inhibit the generation of an action potential, thus the inhibitory, right? Right. And so, right, each of these in natural circumstances have a role to play. It's not that if it's inhibition that's happening, then it's a bad thing. No. So each of these have their role to play. So obviously, action potential not generated. Now, once the transmitter has done its job, you don't want it left bound to the receptors, because that will either cause a continuous excitation or continuous inhibition, and that's also a bad thing. So, right, after the transmitter has had its effect, it is broken down by enzymes, right, and that frees up the receptors, and then right, they are moved back to, for example, the presynaptic neuron, where they are recycled. So they're not wasted, they are recycled. So they're used again. So whatever products they're broken up into, right, they're taken back up, they're rejoined to make the neurotransmitter molecule. One thing to highlight, right, is that the junction between a motor neuron and a muscle fiber, right, or muscle cells, is called a coming from neuron, muscular junction, right? And if an impulse reaching the muscle, obviously what you would expect is for contraction to happen. Or in case if it's inhibitory, a contraction would not occur, depending on, right, what kind of impulse is coming in. Any questions?
No, sir.
Okay, so with the time we have left, let's briefly talk about neurotransmitters. What kind of neurotransmitters generally do we have in the body? So one of the most common ones is acetylcholine, or ACH, right? And it is made using ATP, right? And so neurons have a very high, uh, density of mitochondria, and the nerves that use acetylcholine are called cholinergic neurons, right? And once it has done its job, so after, you know, its effects, it is broken down by the enzyme acetylcholine esterase, right? And this enzyme is present in the membrane of the postsynaptic neuron. Next to the acetylcholine receptor. So what this does is it breaks up acetylcholine into uh, its components, which are acetate, and choline. And they move to the presynaptic neuron, and are recycled again to make a new acetylcholine. Right, so ACH, or acetylcholine, is used in all motor neurons, and the parasympathetic nervous system, so the parasympathetic nervous system controls functions such as digestion, right? So it's part of the resting state of the body, as opposed to the sympathetic nervous system, which is responsible for fight or flight responses. And so that activates the body, so it raises your heartbeat and blood pressure and everything, whereas the parasympathetic brings everything down. It's the resting, right, uh, nervous system, let's say. And both of those are part of the autonomic nervous system, which basically means, right, all the processes in the body that work involuntarily. So autonomic. Okay. There are others. That have other neurotransmitters. So you have noradrenaline, right? And it is used by neurons in the sympathetic nervous system that I just mentioned. And the neurons that use these are called adrenergic. So adrenergic neurons or nerves. And so what they do is, right, they bind to the receptor, and yeah, and everything else that's mentioned about these are, pretty much the same. So they bind, they are used, they're taken back, they diffuse, all of that. So yeah, that covers it. Now there's one thing left. In fact, I'll cover it quickly. If you have, like, we do have a few minutes of class, and right, that is chapter 8A4. So are you still with me?
Yes, sir.
Okay. So effects of drugs on the nervous system. Right? And so by definition, a drug, right, is any chemical or substance. So a drug is a chemical substance that produces a biological effect or causes a biological effect. And so it's not just antibiotics, which kill bacteria, which is a biological effect, but they can be drugs to control the heart rate, for example. So they're bringing down blood pressure, or they're slowing down your pulse, or they are acting on the absorption of some ions or molecules. And so all of those are drugs, right? And they are drugs that affect the nervous system. So an example is having an effect on the blood pressure. So in general, there are two types. One, right, we're talking about, right, the two types of drugs that we are talking about are ones that increase an effect. So they're excitatory drugs. So they increase the response of the nervous system. And so what they do is they increase the amount of neurotransmitter that is synthesized and released. And those neurotransmitters obviously bind to receptors on the postsynaptic neuron and activate them. and that what they also do is, because it's a drug, it prevents the degradation of the neurotransmitter, or prevents the reuptake of the neurotransmitter into the presynaptic knob. So the drug prevents the breakdown of the neurotransmitter or the reuptake of neurotransmitter into the presynaptic knob. All right. And so because it's not letting the neurotransmitter let go of the receptor, and so it increases the response that, as compared to what you normally have. The other one, as you can imagine, is causing an inhibition, so it is decreasing the response. So those are the other kinds of drugs. And again, by drugs, right, we're not talking about, illegal drugs, painkillers. So, they work by decreasing responses, right? Or anti-inflammatory drugs, NSAIDs, right? The Panadol that you take to, reduce fever, or when you're having a headache, those are decreasing responses. And so how they work is that they block the synthesis of neurotransmitters, right? And acha, in fact, let me write this out. They cause the neurotransmitters to leak from the vesicles and be destroyed by enzymes within the fiber. Because the neurotransmitters are meant to be released in the synaptic cleft, so if they're released within the fiber, there are enzymes in the fiber that will digest them. I'm using the term digested, or they're broken down by the enzymes. Then they prevent the release of the neurotransmitters. And finally, they block the receptors and prevent the neurotransmitters from binding. We just go back. And put this in as a separate point. So they increase amount of neurotransmitter released. And so with that clarification now, to right, drugs don't do all of these. So drugs work via any of these mechanisms. So now they're not doing all of it, that a single drug that's decreasing response will block the synthesis and cause them to leak and prevent them from release and block. They're not doing all of them. So it will work on its mode of function. Will be any one of these. And similarly, right, for the drugs that cause an increase in the response, they'll either increase the amount of the neurotransmitter that is being synthesized, or the amount that is being released, or the binding and activation of the receptors, or they prevent its breakdown. So any one of these, not all of them at the same time, just to be clear. Now, what's given after this in the text is something I leave to you as homework. They're mentioning 3 different drugs, right? And those are nicotine, that is inhibitory, that is excitatory, right? So it is, and then you've got lidocaine, and then you have cobra venom, yeah, alpha cobra toxin. So alpha cobra toxin. And so they just mention how they work. So the, nicotine is a stimulant, right? And triggering action potentials. Lidocaine is an anesthetic, so it is decreasing, it is blocking the action potentials. And then cobra venom is also toxic. It affects neuromuscular junctions and inhibits muscular contraction, which can be lethal if it affects the brain and the lungs and everything. But in low doses, it can be helpful. So, right, I just want you to read that on your own. There's nothing technical there. And with that, we are done with the chapter of talking about the nervous system. What's up next is the sensory systems and detection of light. So we're going to talk about the eye. I'll see whether that's the next chapter that we do, or we move on to something else. We can come back to it later. But in any case, right, so we're done with the nervous system, and yeah, we're done for today. Any questions? I hope everything's clear.
Yes, sir, it is.
Okay, so have a good weekend, and I'll see you next time, inshallah.
Have a good. Chalo, sir.